In the previous lecture, we talked a bit about the evolution of the brain and how we went from very simple, humble beginnings to gradually having what would a structure that towards in the head, which can be referred to as a brain, and that can be a cerebral ganglion, say in an insect, or it could be the brain that you would see in a human. Now, if we look at animals that are quadrupeds, animals that are derived from fish, such as birds, reptiles, and mammals, only mammals have a structure called a neocortex. So neo means new, and cortex is Latin for bark. And so this is a new structure that's sort of laid down on top of, it physically lies above some of these older brain structures that are found in fish, birds, lizards, and so forth. And so this neocortex is something that only mammals have. In humans, for example, here we can see in this picture, the neocortex can be divided into 4 lobes: the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And it's thought that this, the neocortex actually allows for higher order brain functions, such as sensory perception, cognition, generation motor commands, spatial reasoning, and language. So a lot of the things that make us human are derived from having this structure called the neocortex. And interestingly, of course, animals that don't have the neocortex do some of the similar sorts of processing. They just simply do it with older brain structures. And we also, as humans, continue to retain some of these older brain structures. They're buried a little bit deeper within the brain, but they're still there. So anyway, this is a very important structure, and we'll talk quite a bit about the neocortex next because of its importance, particularly for, as I said, higher cognitive type functions that we see in humans. Of course, there's a cost to having a neocortex and a large brain, which is brains are very costly in terms of energy use. So at rest, in a human, the human brain consumes about 20% of the total energy used by the body. In all vertebrates, that is, animals with backbones, there are 2 major divisions of the nervous system. There's the central nervous system and then the peripheral nervous system. So, for example, the central nervous system consists of itself of 2 parts, the brain and the spinal cord. And then the peripheral nervous system consists of all the nerves that lie outside of the central nervous system that help to connect the central nervous system to limbs and organs. In order for us to understand some of the modern theories of how the brain works, which we'll discuss in a second, it's instructive and interesting to look at how people thought the brain worked in the past. So we'll look at a couple early theories about how the brain works. One idea was the aggregate field view. According to this view, all the areas of the brain participate in every cognitive function. So this idea came out of the work of Jean-Pierre Flourens. He was actually very instrumental and very important. What he did was he demonstrated that the main divisions of the brain are responsible for largely different functions. So he did find that out. He found by doing studies on animals that different parts of the brain did have different functions. However, when he looked at cognitive functions, things that are sort of considered higher cognitive functions like memory and thought and things like that, he wasn't able to find any specific regions for those functions. And so this led him to then believe that, well, these sorts of functions must be represented in a diffuse manner, meaning it's spread out. So it's represented in a diffuse form around the brain. And this is referred to as the aggregate field view. The aggregate field view of how the brain worked was probably proposed by Jean-Pierre Flourens in part because of the limited experimental methods available to him in the 1800s. These methods were not sensitive enough to detect localized processing in different brain regions. We now know that the brain is modular, meaning that different parts of the brain are specialized for processing different types of information. We also know that these modules are highly interconnected, allowing us to perceive the world in an integrated manner. A good example of this would be when we look at a painting, such as this one by Paul Cézanne, We perceive these objects as apples. We don't see them simply as color and shape. We see, but the way our brain actually works, it actually processes the color and the shape separately. But then, due to this integration, this highly interconnected nature of the way the brain works, we perceive the colors and the shapes together, and we see apples. Interestingly, another early theory of how the brain works, which is phrenology, was very much the opposite of the aggregate field view. The phrenologists believed that personality could be read by feeling the contours of the skull. The idea was that as the brain develops and grows, it pushes the skull upward. Therefore, there should be an elevation in the skull for areas of the brain that are well developed. So, for example, if we look at this picture, a person who's particularly cautious would have a little bit of a bump there on the side of their skull. The belief that you could predict the personality of a person simply by measuring the bumps on their head now seems laughable. But for a long time, phrenology was treated as science. We now know that it's actually pseudoscience. Pseudoscience is a collection of beliefs or practices that are claimed to be scientific, but are actually not based on experimental evidence. When people actually did do experiments to see if there was anything to this idea, it was largely discredited. There was really no evidence that you could measure a bump on a person's head and then figure out their personality. Shown here is a picture of Franz Joseph Gall, who was a pioneer in the study of the localization of mental functions in the brain, and he's often thought of as the founder of phrenology. Now, the crazy interesting thing out of all of this is that despite the insane craziness of this phrenology idea, is that there is actually some truth behind the idea. So Franz Joseph Gall was correct in the fact that many mental functions are in fact localized within the brain. To understand the brain, we need to understand its component parts. The brain is made up of two kinds of cells: nerve cells, which are also called neurons, and glial cells, also called glia. Neurons are the cells that process information and send electrical signals over long distances. Glia are supporting cells that help neurons function properly. So, for example, one function of certain glial cells is to help regulate the chemical environment around neurons. There are three basic types of neurons. Sensory neurons detect information about the external and internal environment. Motor neurons stimulate muscles to contract. Other output neurons are similar to motor neurons in that they send information and they stimulate organs and glands. Finally, the vast majority of neurons within the human body are interneurons. These are neurons that lie between the sensory neurons and motor neurons. So in other words, interneurons don't communicate directly with sensory receptors, and they don't communicate directly with muscles or glands or organs. They lie in between. Interneurons are involved in much of the computations that are done by the nervous system. And one type of interneuron we may talk about in the future are called projection neurons. Projection neurons are interneurons that transfer information from one part of the brain to another. In this picture, we see a Purkinje cell, which is a type of neuron that's found in the cerebellum. And this type of neuron is involved in controlling motor movement. And you can see it's incredibly pretty. It has many, many branches. And so you can see that the individual neurons may be very complex. Having said that, much of the complexity of the nervous system actually arises not so much from the individual neurons, but it's due to the level of interconnectivity between neurons. So as I mentioned before, the current best estimate of the number of neurons in the human brain is 86 billion. For our purposes here, let's just round that up to 100 billion. And let's assume that each neuron receives about 10,000 connections from other neurons. So if this is the case, then there would be about 1 quadrillion connections in the human brain. Now, Perhaps the number is a bit lower, perhaps it's a bit higher. We don't really know for sure. But this is a gigantic number, and it does give you some idea of the vast complexity of the human brain. Despite this incredible complexity of the human brain, there has been a tremendous amount of progress in understanding how the brain works. And one feature of the brain that helps us with trying to understand it is the fact that the brain has many repeated anatomical structures. In other words, when we look at the brain, we'll find repeated motifs, repeated anatomical structures that will arise over and over again. And this actually helps us because if we can understand how a little piece of the brain works, we may be able to extrapolate our knowledge to other parts of the brain. Shown here is a picture of the neocortex. You can think of the neocortex as a highly folded sheet of neurons. So it's quite large if you were to just spread it out as a large sheet. And so in order to fit this sheet within the skull, it has these corrugations. And that's why you have this sort of wrinkled and grooved appearance of the human brain. Now, this neocortical sheet has an overall thickness, average thickness of about 2.5 millimeters. Now, if we zoom in on this neocortex, we'll see that if we look at a little piece of it and we zoom in, we'll see that there are these things called mini columns. Each mini column is about the diameter of a human hair, and they're about 30 to 40 times longer than they are wide. And what a mini column is it's a neural circuit. In other words, it's a number of neurons, and it's stereotypical. In other words, we'll see the same types of neurons over and over again, and they'll make similar connections from one mini column to another. In other words, there's a lot of similarity between the different mini columns. And we think that these mini columns are sort of a processing unit. They're like a mini CPU within your brain. But instead of having, one CPU or 4 CPUs or 8 CPUs, we have probably millions of these things. And so this gives you some idea of the sort of computational power of the human brain. Despite the similarity of the mini columns all across the neocortex, when we look at different parts of the neocortex, we find that they're specialized for different functions. So, for example, part of the neocortex may be specialized for receiving and processing visual input. Another area may be specialized for receiving and processing auditory input. And so again, this is a very interesting idea that we have essentially the same anatomical structure, this neocortex, which is based on this idea of mini columns, which are little mini processing units. And yet the type of processing they do is very different depending on the part of the brain. So that this means that this structure, the basic anatomical structure of the neocortex is very flexible. It can do different types of information processing. If we look at our nervous system, another place where we see repeated anatomical units is within the spinal cord. But to understand that, we should take a step back and understand a little bit more about how humans are built. So if we look at an earthworm, we can see that it's a segmented animal, meaning that it has anatomically identical units running along the longitudinal axis from the front to rear of its body. And this is also the case of other annelids, which include things like leeches and marine worms. Now, if we look at a different class of animals, the arthropods, this includes things like centipedes, insects, spiders, scorpions, and crustaceans, you'll find that they're also segmented. So you can see the segmentation is very clear in this picture of a centipede. Each segment, for example, has a pair of legs. Now, it turns out that humans are also segmented, as we'll see in the next slide. Most people are probably horrified to find out that they have anything in common with an earthworm or a centipede. But in fact, we do have a lot in common with them. Humans are segmented animals, which is always a bit surprising because if you look at a human from the outside, you don't really see lots of evidence, much clear evidence that we're segmented. But if you do look inside the human body, you see that we are truly segmented animals. And this is most clear if we look at the vertebral column. So we have a series of vertebrae or bones that make up our backbone. And within that, and associated with it, is the spinal cord. So the spinal cord is part of the central nervous system. Remember, the central nervous system consists of the brain and spinal cord. So the spinal cord is also segmented. So for each segment, vertebral segment, there are also a pairs of nerves that come off the spinal cord. So humans are truly segmented animals. And so again, we have, just like we saw within the cortex, where we had repeated anatomical structures, within the spinal cord, we have repeated anatomical structures. We have all these essentially segments within the spinal cord. And that's helpful also. It helps us when we're trying to understand how the spinal cord works. It gives us some insight into how it works. So it turns out that segmentation is a very old characteristic. The reason that we are segmented and worms are segmented and centipedes are segmented, all these very different types of animals are segmented, is that we all had a common ancestor that was segmented and lived probably about 600 million years ago. And we know this through studies looking at genes. So we have some of the same or very similar segmentation genes as these other animals. Just as the cerebral cortex has repeated anatomical units in these mini columns, and the spinal cord has repeated anatomical units in the segments, the cerebellum is also made up of repeated neural circuits. The cerebellum is vital for coordinating movement and learning motor skills. It's a part of the brain that lies here below the cerebral cortex and behind the brain stem. And so if you look at the cerebellum in detail anatomically, you'll find that it has these repeated neural circuits. We've discussed a bit the idea that there's local processing within mini columns, and these mini columns may form modules, which are neural circuits that are specialized for certain types of processing within the neocortex. But we need to discuss how information can get from one of these modules to another. Well, it turns out the neurons have two kinds of branches. They have dendrites. Dendrites normally receive input from other neurons. So for example, shown here, we can see a cell body with a nucleus, and there are these branches. So these are called dendrites, and this would be the site of input from, say, another neuron. Now, many neurons also have another process, which is very long, and this is called an axon. An axon allows for long-distance communication from, say, one cortical area to another. The long-distance communication is achieved by electrical signals, which travel very rapidly from one region of the cortex to another, or also potentially between the cortex and, say, the spinal cord, or from a neuron in the spinal cord out to the periphery to, say, a muscle. The conduction of these electrical signals is aided by what's called myelin wrapping. This is a kind of wrapping that's produced by glial cells. Recall that glial cells are support cells. So for example, in this picture here, we see there's a specific type of glial cell called a Schwann cell, which is found in the peripheral nervous system, which we'll talk about more later. But it wraps around the axon at various points. So you now have a myelin sheath around the axon, and that will greatly increase the speed of the conduction of this electrical signal. Now, it turns out that the myelin consists of essentially fatty substances. It's lipids, which is a kind of fat. And as you know, fat has a white appearance. So in a region of the brain where there are a lot of axons that are myelinated, you'll see that the brain has a lighter or white appearance. Anatomists discovered very early on that when you slice through a brain, whether it be a human brain or a sheep brain, or some other mammal, is that there is a distinction between in color between different parts of the brain. The cortical regions, the areas that are essentially form the bark on the outside of the brain, tend to have gray matter, meaning that they're grayish in appearance. Whereas if you look a bit deeper, the brain has a whitish appearance. And we know now why that's so. The white matter consists of what are called fiber tracts. Tracts are bundles of axons, and these axons are myelinated, meaning they're wrapped in myelin, which is a fatty substance, and that gives this white matter its whitish color. In the gray matter, there may be some axons that are myelinated, but the majority of the axons aren't myelinated. And the reason for this is because the axons don't have to send nerve impulses quite as fast, because they're doing local processing over very short distances between neurons that happen to be neighbors or near neighbors. So you have a lot of cell bodies, you have glial cells, and you do have axons, the majority of which are not myelinated. And that gives you the grayish appearance of the cortex. Now, if you look outside of the brain, there are also bundles of axons, and these are called nerves, and we'll talk about those more later. To summarize, we've talked quite a bit about the idea that the nervous system is organized into modules. Each module consists of many neurons that work together to perform a computation on inputs and then send the results to other modules via projection neurons. Each of these modules will perform a specific function. So, for example, you may have a module that's involved in hearing a certain frequency. You might have another module that's involved in a certain type of processing and visual input. And normally, there's such almost seamless coordination of the activity of these modules and interaction between these modules that when you say you have a visual field and you see something like a mouse, you don't just see gray and you don't see mouse shape as separate things. You see, well, there's a mouse sitting there looking back at me. And so that occurs when you're conscious, when you're able to, when all these modules are interacting in an appropriate fashion. Now, there are, of course, medical situations in which you would prefer that a person not be conscious. And so when a person goes into surgery, they go under general anesthesia. And this doesn't turn off the brain. The various modules are most likely pretty much just as active as they would be normally. But what's happened is there's been a disruption of the communication between the modules. And this renders a person unconscious, and it also makes it very difficult for them to form memories, which is probably a good thing. You wouldn't want to remember exactly what happened during, say, surgery.